The speaker of today is Dr. Junji Iwahara from University of Texas Medical Branch. Dr. Iwahara got his PhD degree in biophysics and biochemistry from University of Tokyo in 1998. He then moved to the United States and did postdoctoral work on NMR biophysics at the UCLA and NIH. In 2007, he started his own lab at the University of Texas Medical Branch, where he is now professor. The title of the presentation of today will be Quantifying and Visualizing Weak Interactions Between Anions and Protein, a topic which is of great interest, has already been touched several times. So I'm very happy to have Junji here, and please go ahead. Thank you.
Great, thank you. Thank you for the introduction. Let me start screen sharing. Can you see my screen now?
Yes.
Okay. First of all, I would like to thank Walter and other organizers for this opportunity. This is really a great opportunity for me to share our recent findings with others. I changed the title a little bit. The original title was Quantifying and Visualizing Weak Interactions Between Anions and Proteins. But now the updated title is different in the last phrase. It's between ions and biomolecules, because we study both cations and anions, and we study both proteins and nucleic acids. We are particularly interested in DNA recognition by proteins. As you know, DNA is highly negatively charged, and the molecular interface of proteins, DNA binding proteins, is positively charged. And when they make complex, electrostatic interactions play a major role. The contribution of electrostatic interactions to binding free energy is in both enthalpic terms and entropic terms. Of course, the electrostatic interactions cause decrease in enthalpy, and also by counter-ion release, the electrostatic interactions also make the major contribution in entropic change, because when proteins bind to DNA, ions condensed around DNA are released. So this release of ions basically is supposed to cause more freedom for ions, and it contributes to an increase in entropy. So to understand thermodynamic contributions by these effects, we have to know the behavior of ions around DNA and around proteins. And to understand such ions, oops, doesn't move. Sorry, there is a problem. Okay, let me use this one. Oh, now I can switch the slide. Okay, I should first introduce the concept of ion atmosphere. Okay, the ion atmosphere is a zone where the ion densities are different from background. Here are two parameters, delta N anion and delta N cations, parameters called ion excess. And this is the difference between the ion density in the background and ion density in the ion atmosphere. It corresponds to the number of accumulated or excluded ions in the ion atmosphere. And the charge neutralization occurs through both counter-ion accumulation and co-ion exclusion. For nucleic acid, for DNA particularly, counter-ion condensation theory is quite popular. This is quite a simple theory. And just from the parameter of charge spacing, you can predict the ion excess for counter ions and core ions. But this theory is not applicable for globular systems like proteins. And also, this is not applicable for mixed charge systems like proteins. For more general charge distribution and molecular shape, you can use, of course, Poisson-Boltzmann theory. Poisson-Boltzmann equation is about electrostatic potentials and also Boltzmann distribution of mobile ions. And using statistical weight of Boltzmann factor together with subtraction background, you can calculate ion excess for anions and cations separately based on this theory. But Poisson-Boltzmann theory is not perfect. It is an approximate theory, especially for ion distribution near the surface could be not very accurate. So some people use a different theory to predict ion probability distribution around proteins and DNA using different models. For example, this is a figure from Monty Pett group's paper published in 2011. They used integral equation theory and applied it to some different DNAs. And here they showed probability distribution for potassium ions and chloride ions. And they found some difference between different DNA sequences in terms of ion distribution. Some people also use MD to study ion distribution around nucleic acids. And this is from a paper published in 2015. It was from Richard Raveri's group. They use long MD to analyze ion distribution around DNA. But depending on what kind of models, theoretical models you use, you may get quite different results. It was nicely demonstrated by this group of David Case and Darren York. They compared ion distribution, spatial distribution around the 24 base pair DNA in this paper. And this is a 2D plot of the ion distribution. And this 2D plane is perpendicular to DNA axis. So what you are seeing in shadow here is actually Watson-Crick base pair. And here, this is ion distribution from MD, and this is from 3D rhythm, and this is from non-linear Poisson-Boltzmann equations. And as you can see, the ion distributions are quite different for these results. Given this kind of results, of course, we have a question. What are really realistic view? I mean, what is more realistic distribution for the ions around nucleic acid or proteins? Of course, to answer the question, we have to use experimental data. But when such experimental examination is quite difficult, we experimentalists feel like this. We are far behind theoreticians. But in ideal situation, we experimentalists can come up with a technology, and with the technology, both experimentalists and theoreticians can make progress together. That's an ideal situation. And this webinar series is really ideal for that. I mean, it is an ideal catalyst for that because both theoretical people and experimental people are joining and discuss science. For example, last month, Franz Mulder presented nice NMR modern technology to measure pKa accurately. Earlier in this month, Thomas Lowy showed analytical electrophoretic methods to study protein charge. What is my technology? Okay, my technology is also NMR based, but it is more about ions. In some experiments, we use NMR to directly observe ions around biomolecules. In other experiments, we observe protein or DNA NMR to study ion macromolecular interactions. And by having these two from different perspectives, hopefully we can get better understanding of ion protein interaction or ion nucleic acid interactions and their role in protein DNA association. Today, I'm going to introduce some of our recent results from the work published in 2 papers. This is about characterization of ion diffusion around proteins DNA and also protein DNA complex, and quantifying and visualizing ions around those molecules. Okay, there is a long history about the ion atmosphere around DNA. Even in 1970s, people used sodium NMR, sodium 23 NMR relaxation to study interactions between sodium ion and DNA. But quadrupole relaxation is quite difficult to interpret in terms of physical properties of ions. And in early 21st century, Louis Pollack's group used anomalous small angle X-ray scattering to detect the physical presence of ion atmosphere using heavy alkali metal ions like rubidium or cesium. More recently, Dan Harshrak's group developed the method called ion counting method. And they used atomic emission spectroscopy or ICP mass spectrometry to quantify ions around DNA or RNA. And the ion counting method was really helpful to advance the field of ion atmosphere around nucleic acid. But these methods are not really perfect. They are not suitable for investigating, for example, dynamic properties of counter ions and a release of counter ions upon protein DNA association. So there is quite a lot of room for methodological development in this field. Today, I'm going to talk a lot about ion diffusion. So let me briefly explain how the method works. Okay, of course, diffusion is about diffusion in liquid. So we use liquid NMR, and this is an NMR sample. And we typically use a uniform magnetic field. In other words, at every point throughout the NMR sample, the NMR frequency is identical. But we can introduce magnetic field gradient, and then the resonance frequency becomes dependent on the position. Okay? And at this point, basically, the coherence is decorrelated, and it is what we call Dephase, it's NMR jargon. And here, we can rotate nuclear magnetizations by 180 degrees and apply the same magnetic field gradient again. And then something magical happens, and dephased magnetization get regain coherence. This phenomenon is called rephase. But this rephase is not perfect because of diffusion. For example, if one spin was located here during the D phase stage, but through diffusion, it moved to the position right here, for example, then the frequencies during D phase stage and during a rephase stage are different. And because of that, the rephase doesn't become perfect. Of course, this effect is stronger for faster diffusion and weaker for slower diffusion. And this effect also depends on the magnitude of the gradient. So by analyzing this rephasing effect as a function of gradient strength, we can determine a diffusion coefficient for ions or macromolecular components or other small molecules. And we use ion diffusion to study ion condensation and counter-ion release. Basically, ion condensation should make the apparent diffusion slower. In other words, a diffusion coefficient should become smaller, and counter-ion release should cause increase in the apparent diffusion coefficient. In some experiments, we use, for example, this buffer. Okay, this buffer contains 20 millimolar N15 labeled ammonium ions and N15 labeled formamide. And macromolecular components are not labeled. So if you measure nitrogen spectrum, you get a beautifully simple spectrum like this one. Only 2 signals are present. Usually in protein animal spectroscopy, we see hundreds of signals, but here we see just 2 signals. And we can measure diffusion for this. And we can use this approach to study ion condensation and counter-ion release. Okay, here is an example. This is showing diffusion coefficients of ammonium ions and formamide as a neutral internal control. And the coefficients were measured at some different concentrations of Antennaepedia homeodomain protein. In this case, the diffusion coefficients are almost identical, independent of protein concentration. But if you look at closely, actually there is slight decrease. It's due to macromolecular clotting effect. If you do the same experiments for DNA, the results are quite different, like this, okay? The diffusion of ammonium ions become significantly smaller when DNA concentrations increase. This is due to ion condensation. But to confirm that this is really due to ion condensation, we did an assay called, we call ionic competition experiment. And in this experiment, for example, we took this data point, a sample of this data point, and added KCl to the sample, and then observed behavior of the ammonium ions and formamide. For the formamide, it was not affected by KCl concentration, but ammonium ions diffusion became faster. It's due to competition between potassium ions and ammonium ions. Basically, when KCl concentration increases, the population of ammonium ions within ion atmosphere decreases. And as a result, the diffusion coefficient becomes larger. So this decrease as a function of DNA concentration is due to ion condensation around DNA. And we can analyze this concentration dependence to get diffusion coefficients of ammonium ions within ion atmosphere. Okay, so this is diffusion coefficient of ammonium ions within the ion atmosphere. And quite importantly, this diffusion coefficient within the ion atmosphere is still 10 times bigger than the diffusion coefficient of DNA molecule itself. Basically, if all the ions are moving together with DNA through tight binding, the diffusion coefficient for ion atmosphere should be identical to the diffusion coefficient of DNA, but that's not what we are seeing. We are still seeing quite a high degree of mobility of ions, cations, in ion atmosphere. We can do the same experiment for other metal ions. Quite recently, actually only a few months ago, we were able to purchase and install diffusion broadband probe capable of generating a much stronger gradient and detecting various different kinds of nuclei. This probe can generate about 5 times stronger gradient than typical probes, and that facilitates our diffusion measurements. And using this, we were able to do the same experiments for sodium ions, and here is the results. This is basically the same set of experiments for sodium ions. And again, here we see decrease in diffusion for sodium ions when DNA concentration is increased. And here we see increase in the diffusion coefficient of sodium ions when KCl was added. And again, we can determine the diffusion coefficient for sodium ions within the ion atmosphere. Now, we are even trying to get another diffusion probe, custom-made diffusion probe, that is capable of producing an even stronger gradient to observe chloride ions, because this probe cannot observe chloride ions. And diffusion measurements of chloride ions requires much stronger gradient due to rapid relaxation and basically a small gyromagnetic ratio. But until we get the new probe, we can still design NMR experiments to study anion protein interactions. For example, we can use carbon-13 labeled acetate ions, which is commercially available. And we can make this kind of buffer containing carbon-13 labeled acetate and carbon-13 labeled DMSO. This DMSO is just a neutral internal control. It's just a little bit, just a 10 millimolar. We can do the same experiments for protein solutions to see the anion protein interactions. We did this kind of experiments for some different proteins, and 2 of them are shown here. One is Antennaepedia homeodomain, and the overall charge is positive 12 for this protein. Ubiquitin is with 11 positively charged residues and 11 negatively charged residues, and as a result, the total charge is 0 for this one. Okay, for these 2 proteins, we did same diffusion experiments, but this time for carbon-13 acetate, and we got these results. Okay, this is a protein concentration dependence data, and green is for ubiquitin solutions at different concentrations, and black is Antennaepedia solutions at different concentrations. Here, we did see quite a significant decrease in acetate diffusion for Antennaepedia homeodomain. But this decrease was quite small. Actually, here, this point is not 0. Real 0 is down here. Okay, so this difference is much smaller than what we observed for DNA solutions, reflecting weaker interactions. of course, DNA's charge is much larger than a protein's charge. So impact is smaller. But when we did ion competition experiments, adding KCl, the diffusion of acetate become larger. So this assures us that what we see here is really due to basically ion-protein interactions. But even for DMSO, in this case, there is a decrease, right? And this is, again, macromolecular crowding effect. So for anion protein interactions, we should really take this macromolecular crowding effect into consideration. With that, we can still determine the diffusion coefficient for acetate ions within the ion atmosphere around the protein. That's what we got here. Okay, and for this one, the diffusion coefficient for the acetate ions in the free state and in the bound state, I mean, within ion atmosphere, are not very different. Probably due to relatively weak interactions between positive charge of protein and acetate. But still, the decrease was significant, and there is some degree of restriction of diffusion. That's what our experiment data show. We can also use this method to quantify anions around proteins. And here, demonstrate how it works. Okay, when we equilibrate protein solutions with this buffer, actual signal intensity of acetate ions depended on the protein concentration. The reason is quite simple. Basically, here, the concentration of protein is millimolar range, and concentration of acetate in buffer is 20 millimolar. So when protein accumulate ions, actually those accumulated ions can contribute to the increase in the overall concentration of acetate. You know, the ions bound to macromolecules cannot go through the membrane used for buffer equilibration. So the overall concentration of ions becomes higher. Using this, we can measure actual ion excess for the ions around ion atmosphere of these positively charged proteins. Yeah, this is concentrations of acetate measured in this way at different concentrations of Antennaepedia, BPTI, and ubiquitin. BPTI is also small protein, but with positive 6 charge. And for those proteins, the concentration dependence here can give us the number of acetate ions per protein. The slope here gives the number of acetate ions per protein. And as I mentioned, the buffer is designed so that the acetate is the only anion in the buffer. So basically, just by measuring slope here, we can basically measure ion excess within the ion atmosphere. That's what we did. Okay, here. And for Antennaepedia, it was about 7. And for BPTI, it was about 4. When we started this experiment initially, we were expecting to see 12 here for Antennaepedia and 6 here for BPTI, simply because they are positive 12 and positive 6. But our data clearly indicated that it is not the case. Initially, we were puzzled, but it turned out that the answer for this discrepancy is quite simple. Basically, charge neutralization of macromolecules happen through two different things. One is, of course, counter-ion accumulation, but the other one is co-ion exclusion. And this can also contribute to the charge neutralization. And a condition required for charge neutralization is shown here. Okay, so it's not just about counter-ion accumulation, but also about co-ion exclusion. Okay, we used Poisson-Boltzmann theory to calculate ion excess from the protein structures. We used APPS and Delphi, and they gave almost the same, indistinguishable results. And these are basically based on the calculated electrostatic potentials together with integration shown here. Okay, and the theory predicts that the anion accumulation for BPTI is about 3.5, and for Antennaepedia it's 8.2. It's still smaller than the overall charge. And co-ion exclusion, cation exclusion in this case, is quite significant like this. Yeah, that's why this number is not really the same as the overall charge here. This is maybe quite obvious for theoretical people, but for us at least, it was really eye-opening finding, because we were expecting just neutralization by counter ions. But neutralization is also about exclusion of co-ions.
Excuse me, Junji,
yes.
May I ask you a clarification?
Yes.
At which distance from the surface of the molecule you stop counting the counter ion, the co-ions for counter ions?
For this calculation, we used quite wide space. It was, if I remember correctly, like 400 angstroms.
Ah, wow.
So it's, cube size was quite big compared to the size of the molecule.
Okay, so you counted over all the cube?
That's right.
Okay.
All right, and you might wonder why there is error bar for theoretical calculations here. The answer is quite simple. We use structural ensembles generated by basically randomization of sidechain conformers under inference of a conformational database. For regions, crystal structures do not have, we also randomize the structure. And we calculated ion excess for each structure. But actually, this kind of structural fluctuation has very little impact on ion excess. In other words, this ion excess is quite robust, and it's not very sensitive to small details of structures. Okay, now I'm going to talk about counter-ion release. And before I show my data, I should briefly mention about what people did previously. Okay, in 1776, Thomas Record presented a nice theory, simple theory, to estimate number of release counter ions using just binding constants measured at different salt concentrations. And basically, from log-log plot, you can determine the number of released counter ions using the slope of the linear dependence. And this theory is based on counter-ion condensation theory, and quite simple. So many experimentalists use this approach. But theoretical people made a lot of criticism about this approach. It's not surprising because, for example, hydrophobic effects also is affected by changing ionic strength. And even without any condensation of ions at all, the binding constant is still dependent on ionic strength. For example, because the kinetic rate constant for association is affected by screening effect. So this was quite controversial method, although it's simple and really liked by many experimentalists. So we really have to validate, consider whether or not this kind of evaluation is really accurate for estimating the number of released counter ions. For that, of course, the best way is to directly observe counter ions. Okay, this method is not direct observation, right? This is just observing equilibrium constants for protein DNA association at different salt concentration, and nothing is really directly related to ions. So by observing directly the ions behavior, we thought that we can better assess this ion release. That's what we did. Okay, using the diffusion method, we basically measured the number of released counter ions. In this experiment, we measured diffusion coefficient of ammonium ions in the solution of protein DNA complex and in the solution of DNA solutions and protein solutions. And from that, we estimated the number of released counter ions. Of course, this is based on a direct observation of ions, and it turned out to be about 11 cations released upon protein DNA association. And for this kind of argument, people used number of intermolecular ion pairs between protein and DNA. But as I mentioned, it's not really great way. But anyway, let me do the same kind of evaluation here. Okay, in the crystal structure of Antennaepedia protein complex, there are 9 ion pairs between arginine or lysine, cytosine, and DNA phosphates. And the crystal structure doesn't include the disordered regions of N-terminal region of Antennaepedia, but our animal data clearly suggests that at least one of the arginine residue in the disordered region is involved in protein DNA association. So this data, this kind of argument predicts 10 ion pairs, but these 10 ion pairs could be smaller if you use different criterion for the distance. And also, this argument doesn't tell anything about ion release from the protein. So we did experimental estimation of the release of anions from protein. And it turned out that all anions, all 6.7 anions accumulated around Antennaepedia are released upon protein DNA complex formation. In other words, our experiment data showed that 6.7 anions and 10.7 cations are released upon protein DNA interactions. But KD data, binding constant data at different salt concentrations predicted 6.9 ion release. So there is a huge discrepancy here. So we examined whether or not this is reasonable. And for that, again, we used Poisson-Boltzmann equations. And for free protein, free DNA, and the complex, we estimated basically ion excess numbers. And it turned out that basically all anions around the homeodomain, Antennaepedia, must be released. And for the DNA, basically, it released 10 anions. So basically, our experimental observation is quite consistent with Poisson-Boltzmann prediction. Okay, so now we know that more anions and cations are released, and you might expect that larger entropic gain due to larger number of ions released upon complex formation. But probably ion release doesn't make much entropic effects. I mean, it may not be as large as you expect from the number of released ions, because now we know that ions within the ion atmosphere are quite mobile. The diffusion coefficient of the ammonium ions, for example, in the free state and DNA bound state are different only by a factor of 1.7. But if the ions are tightly bound to DNA, the ratio should be like 18. And if you use the theoretical relationship between entropy and diffusion published by Seki and Baguchi in 2015, you expect actually much bigger entropic change for ion release. For the ion release from the protein, the entropic effect is predicted to be even smaller. So the larger number of released ions do not necessarily mean a larger number of entropic gain upon ion release. Okay, now I'm going to talk about not ion NMR, but protein NMR. And we can use protein NMR to study the interaction between anions and proteins. And you might think that, for example, to study acetate protein interaction, you can just add acetate to your NMR sample. But it doesn't work in that way, because if you just add acetate, it may change pH. And if you like to retain the pH, you also have to add something like sodium acetate. And then you also see the impact of cations, right? So we have to design experiments so that the anion is the only difference in different samples. And here we did such a study, and we used this buffer, and the only difference is chloride versus acetate. And when we used these buffers for the free protein, we saw a lot of differences in chemical shift, but not for the complex. A reasonable interpretation of these data is that for the protein, it's positively charged. So the difference in anions can make a huge difference. But for the protein DNA complex, because overall charge is still negative, the attraction of anions are not really significant. And because of that, even if you change the anion from chloride to acetate, the chemical shift differences are quite small. But this is qualitative discussion. And to study ion distribution, you need more quantitative information. For that, we decided to use PRE, paramagnetic relaxation enhancement. And this is distance-dependent quantity caused by dipole-dipole interactions between a nucleus and unpaired electrons. And because of a very strong magnetic moment of unpaired electron, this effect is quite strong. And even if the distance is relatively large, you can still observe that impact. And when I was a postdoc at Marius Kroa's lab at NIH, my theme of research was impact of dynamics on PRE. At that time, we used a PRE arising from unpaired electrons introduced in macromolecules. But we can use unpaired electrons in other molecules, like co-solute, like TEMPO or DTP and gadolinium, or just dioxin, as Franz used in his previous work. So basically, for this type of PRE, large PRE is observed for a proton nuclei near surface, and proton nuclei deep inside of the molecule usually shows a relatively small solvent PRE, or this is called solvent PRE. And we decided to use solvent PRE to study anion protein interactions. And for that, we used this compound, which is analog of acetate ion. And we used this compound as a control, okay? And when anions are accumulated particular region, basically this one is also accumulated. So compared to this compound, it gives a larger PRE. It means basically the difference between these two defined in this way becomes positive. On the other hand, for the region where anions are depleted, the sign of this delta gamma 2 becomes opposite. So in this way, we can identify regions where anions are accumulated or depleted. That's what we did for Antennaepedia, and it's complex with DNA. And this is the data for the free protein. And you saw a lot of positive delta gamma 2 here, which means that these regions accumulate ions. But for the complex, no residues really show significant positive gamma 2. But many residues show negative delta gamma 2. And if you plot the data here, the distribution is like this. Again, here, green is the region where anions are accumulated, and orange is the region where anions are depleted. And if you compare the results with electrostatic potentials, I mean surface electrostatic potentials, you can see quite consistent results. Basically, for the positively charged surface, you see anion accumulation. But when the protein makes a complex with DNA, the protein surface becomes also with negative electrostatic potentials, and that causes basically anion depletion. I should mention that this is also consistent with our diffusion data, showing basically anion release upon protein DNA complex. Okay, here are the conclusions. Basically, NMR-based methods can provide quantitative information about ion atmosphere around biomolecules. And NMR-based diffusion measurements for ions provide a unique insight into ion condensation and release by biomolecules. And NMR PRE data can reveal a spatial distribution of ions around biomolecules. Maybe this cartoon right here best represents the conclusion. Basically, with NMR, we can make progress in protein electrostatics. Okay, these are people who made contributions to this work. Bing Han Yu and Channing Pretka are member of our lab, and Monty Pettit are great theoretical advisor for us. We are collaborating for quite a long time. And Tianzi Wang is a facility manager who maintain our NMR instruments in great shape. And these are funding support. And thank you for your attention.
Thank you, Junji. That was a very interesting presentation and very in line with other talks which has been discussing about ionic distribution around biomolecules. Waiting for other questions, I have actually a few. One of them is you showed at the beginning of your presentation that alternative computational method in order to estimate the ionic distribution can be molecular dynamics and 3D RISM, which, well, molecular dynamics, of course, in order to relax and find the equilibrium distribution of ions is much more time-consuming with respect to Poisson-Boltzmann, 3D RISM is kind of probably a little bit more expensive, but on the same order of magnitude. Did you compare the ionic distribution also with 3D RISM, for example? And can you make some comment on it?
Okay, so for that, yeah, we are now using some different probes for paramagnetic relaxation experiments. And yeah, we recently found that the solvent PRE can be used to estimate even near surface electrostatic potentials. I didn't talk much, I didn't talk anything about that, but we are doing that kind of experiments. And basically, through such experiments, we can learn more about ion distribution. And in the future, we can probably compare the experimental results with some different theoretical predictions. And that's what we, Monty and I are planning, for example, for electrostatic potentials calculated from Poisson-Boltzmann and ion distribution from MD. Basically, as you know, Poisson-Boltzmann is not going to work at very high salt concentration. But ion distribution from MD could be more realistic, even at higher salt concentration. And we are able to run our NMR experiments at relatively high salt concentration. So yeah, we can basically highlight the weakness and effectiveness of some different theoretical models. Now we have a great experimental tools for that. Mm-hmm. We are planning to do such comparison to basically examine weakness and strength of some different theoretical models.
Okay, and the second question is, are you able to say whether the localization of ions are mostly due to electrostatic effects, or can you, or did you also observe specific absorption events?
From our experimental data, the mechanism of condensation is quite difficult to tell. But through ion competition experiments, we can at least compare, relative affinities for different ions. And maybe we can discuss something through comparison of so-called competition coefficient for different ions. For example, some ions have quite high charge density, like lithium ions. Some ions are much weaker in charge density, like cesium ions. And maybe by running some experiments for those different metal ions, we can get some more insight into the mechanism for ion condensation as well.
Okay.
And now we have broadband probe, so we can do a lot of NMR experiments for different metal ions.
Okay, very good. Thank you very much. So I see a question from Franz Mulder, for example. Go ahead.
Yes, Junji, that was a beautiful work. My first question would be if you have a postdoc position for me, I'd like to come to you. I think that would be fantastic. Then I also have a few other questions. So now that you said yes, right? Yes, you said yes. One practical question. So how do you ensure when you start with your protein or DNA that they are actually free of ions? Because you want to have a reference. Don't you need a reference level of making sure that when you do a binding experiment that there's nothing to begin with?
Okay, we use extensive buffer exchange using something like Omicron Ultra. So usually people use those spin concentrator for buffer exchange, right?
Mm-hmm.
And if you do such buffer exchange quite extensively with dilution factor more than, let's say, 10,000 or even more, the buffer equilibration is probably quite complete.
Ah, okay. Yeah, that sounds, I mean, given the other results that you presented about the fact that the affinities are relatively weak, that I guess that would make sense. I was wondering if it would have been necessary to use electric fields or something like that, but it's apparently not at all necessary.
Yeah, of course, for some tight site binding, not territorial binding, maybe buffer exchange is not really efficient for completely remove such a tight binder. But ours are not really seeing such tight binding, and everything is in fast exchange. For example, for ammonium ions, we just see one ammonium signal because there is no tight binding at all.
Yeah. Then if I'm allowed, I will ask another question. That is the last part is really exciting for a lot of reasons. So first of all, a technical question again, because of course in the earlier studies with your anions and cations, you were simply looking at sodium or rubidium or something like that. So that's quite clear. But when you use these molecules that have unpaired electrons, these are bulkier and they are also hydrophobic. So especially when you add them to a protein, you might expect that there is also a problem of not just binding on the basis of charge, but also on the basis of other types of associative mechanisms. So is this a problem and can you correct for it?
Yes and no. Okay, so basically, the reason why we need a really appropriate control is exactly the fact that there are some other attraction force like hydrophobic interactions, as you mentioned. And we have to basically extract such effects by comparing the results for the charged proxial derivative and non-charged, or by comparing the results for the positively charged proxial and negatively charged proxial. The comparison is really important to cancel other effects, I mean, effects other than electrostatic interactions. That's a quite important thing. And recently, we are working on basically de novo determination of near surface electrostatic potentials. And for that, we use a positively charged proxial and negatively charged proxial and take a ratio, not actually the difference. And by taking the ratio, we can partially cancel other effects. And in that way, our concern about other effects like hydrophobic interactions becomes less.
Yeah. So this is when I studied physical chemistry, I didn't really know what was the point, but all of it starts to make sense to me now because now with the data that you show, all these things about electrochemical potential, they start to make so much more sense to me. And also what you showed seems to agree with something else that we recently observed, which is that now if you can compute the electrostatic potentials, then of course you can compute also the depletion of anions or cations as you're doing. And one of the effects that we observed is that one of the anions that's going to be depleted in a negative potential is going to be the hydroxide ion. And that is actually going to change the amide hydrogen exchange rates. And we recently observed that is predictable. So you can really see the depletion of hydroxide ion and the fact that this causes hydrogen exchange to slow down.
That's a really interesting idea. Of course, that could be used as another probe for investigating ion depletion or accumulation.
Exactly. Yeah. Yeah, we just submitted it on bioRxiv, so you can find it if you like.
Yeah, I will check it out. Thank you.
Okay, I see a question from Marilyn.
Okay, let's see. I echo everyone else. That's really cool. So I'm just wondering how you can, so two questions. One is, do you see the interesting things when you do pH dependence? So you should be changing the nature of the protein and whether the ions behave in a sense as you expect. And the other question I have is just this size of your ion cloud. I mean, you were saying, 400 angstroms away. So that's quite, it seems like fairly far. And I'm wondering how many Debye lengths that might be, if that length is, salt dependent, if the kinetics, say, interior or slower than do the kinetics as you move away have to come back to the bulk kinetics? And I guess that would kind of lead me to ask, like, how does the kinetics in this ion atmosphere compare with the bulk motion? So I guess my questions are the pH dependence of this. Does it go with the protonation state changes of the protein, and how does this ion cloud vary, its properties vary with distance from the protein?
For the first question, it's easier to answer because
Right.
in solution biophysics, and we can use many different kinds of conditions in terms of a solution condition. Of course, we can change a solution pH quite easily, and in that way, we may be even able to change the populations of ionic state and non-ionic states. That's
Right.
possible. But we haven't tried it. Yeah, that's a quite interesting approach.
Mm-hmm.
For the second question, regarding the kinetic effect and size effect, basically for the size effect, that's the only condition. I mean, 400 angstrom is the only condition we try,
Mm-hmm.
based on earlier work by Harshrad's group. We just
Mm-hmm.
use conditions. So we don't know. I mean, I actually don't know to what extent such a large cubic size is important, honestly. But we can easily check. Yeah, we can just run a PBS calculation or Delphi calculation cubic size. Regarding the kinetic impact, that's quite important thing, because I'm also interested in kinetic aspect of molecular association. And of course, for that, screening effect is quite important. And basically, ionic strength changes screening effect quite dramatically, and it may affect so-called electron steering of macromolecules. So yeah, it is an interesting subject, and yeah, of course, for studying kinetics, maybe NMR is not the best method. Of course, there are many kinetics we can analyze by NMR, for example, for conformational change or some other unimolecular processes are relatively easy to study by NMR. Or something like protein-DNA interactions, probably something like stop flow kinetics using fluorescence could be a better approach. But anyway, there are many questions we can pursue for this, and the ions certainly play a quite major role. So there are many things we should study in the future.
Can I just ask, what is the comparison of the rates, diffusion rates you're seeing in your ion cloud compared with diffusion rates in free solution? Do you know that?
Let me show you just a moment.
Okay.
Okay, so you are talking about probably this one.
Yes. Yeah.
Okay, so basically they are different just by a factor less than 2. That was
Okay.
quite surprising to me initially. I was expecting much bigger difference. But this is not just for ammonium ions. Even for sodium ions, it's a similar situation. And for acetate ions around protein, the difference is even smaller. So ion release is not really going to give a lot of. The ions already have quite large degree of freedom, even when they are bound to molecules.
Interesting. Okay, thank you.
Okay, I see that Tom has a question.
Yes, I was wondering, I wanted to echo everyone else. That was a great talk. I was wondering if it's possible to do the competition studies that you showed the one example of with other anions to see if they fall in the Hofmeister series.
Well, that's a really interesting idea. Yes, it is really possible to do such experiments. And we can even determine competition constant for different ions. So yeah, that's really yeah, feasible, although we haven't tried yet. Thank you for the suggestion.
Thank you.
Okay, let me see if there are questions from the YouTube channel. No. Okay, so if there are no more questions, then I'd like to thank our speaker of today again. Thank you very much for your presentation.
Thank you.
And I would like to remind, so next week we will be back to the usual timing, and the speaker of next time will be Pedro Raiz from University of Lisbon. So thank you. I would like to thank everybody, and see you next week.
Okay, thank you.
Bye-bye.